3D tumour spheroids have replaced the conventional 2D monolayer technique as the gold standard for in vitro cancer research. These models permit the co-culture of multiple cell types which reflect the heterogeneity of the tumour microenvironment and allow the exploitation of spatial interactions.
Advantages of using the hanging droplets method to generate 3D spheroids is the lack of cell-plastic interaction and the ease of studying the interaction between the tumor cells and the non-tumor cells. This technique also provides a reproducible and cost-effective way to model tumor-stromal interaction.
3D tumoroids are valuable preclinical models and are often used as drug screening tool. This method can be applied to study the pharmacological effects of multiple compounds on tumour growth and the surrounding microenvironment.
The process of spheroid formation is user-friendly and requires conventional cell culture lab equipment. Spheroids can be imaged using any benchtop inverted light microscope, and the analysis of spheroid size and shape can be performed using widely available online image analysis software.
To begin, remove HUH7 and HEP3B HCC tumor cell lines and COS7 and LX2 fibroblast cell lines from their storage rack in liquid nitrogen and rapidly defrost them. After defrosting, dilute the thawed cells with 2 milliliters of fresh culture medium. Centrifuge the cells, discard the supernatant, and resuspend the cell pellet in 1 milliliter of fresh water.